We can examine much of the world of biology with our unaided eyes. But without a microscope, we could never experience the world of microbiology. In this lab, we will learn about different kinds of microscopes. Then we will discuss the proper use and care of a compound microscope. A microscope is an instrument that uses one or more lenses to produce a magnified image of a small object. Some of the earliest microscopes were little more than single-lens magnifying glasses. Scientists quickly learned to build better microscopes with multiple lenses and greater magnification. Today, there are many types of microscopes, but all microscopes can be classified as either optical or non-optical microscopes. An optical microscope uses visible light, which passes through one or more glass lenses to magnify an image. A non-optical microscope uses other forms of electromagnetic radiation, such as electron beams, ultraviolet light, or lasers. Magnetic lenses are used instead of glass lenses to magnify an image. An image collector focuses the image, and a computer projects the image onto a video screen. The most common type of optical microscope used in high school and college biology classes is the compound microscope. With a compound microscope, an image is magnified when light passes through a very thin or transparent object and is focused and magnified by a series of lenses. Most student models of compound microscopes can magnify an image 40 to 400 times. But some professional instruments are capable of magnification up to 2,000 times. Another kind of optical microscope is a stereo microscope, which is also called a dissecting microscope. If you recall, a stereo microscope was used during the lab in which we dissected a chicken egg. A stereo microscope uses a series of lenses to produce a three-dimensional image. This type of optical microscope is used to magnify the image of objects that are not transparent. It is also used to magnify objects that are too large to be viewed with a compound microscope. Stereo microscopes can magnify an image up to 40 times. With advances in digital technology, an image of a specimen from an optical microscope can be displayed on a video screen and viewed by more than one person at a time. An optical microscope can be used to view many tiny things, such as bacteria, protozoans, and the cells of plants, animals, or humans. To see even smaller objects, such as a virus, a cell membrane, or the details on the surface of a human hair, a scientist needs a non-optical microscope. Some non-optical microscopes use beams of electrons to magnify images. These are called electron microscopes. Two common electron microscopes are scanning electron microscopes and transmission electron microscopes. A scanning electron microscope bounces a beam of electrons off a specimen to scan its surface. A transmission electron microscope transmits a beam of electrons through a specimen to see its interior features. Some other types of non-optical microscopes include confocal microscopes, which use laser light to magnify images, X-ray microscopes, which use X-rays, and ultraviolet microscopes, which use ultraviolet light. Modern non-optical microscopes can magnify an image of an object up to 10 million times. However, non-optical microscopes are large and extremely expensive, so they are usually found only in large universities and research laboratories. For the remainder of this lab, we will demonstrate the proper use of the compound microscope, which is the type of microscope most commonly used by students. Let's discuss the parts of the compound microscope. The basic parts of the compound microscope are the head, arm, base, nosepiece, and stage. At the top of the head is the eyepiece. The eyepiece is where we look into the microscope to see a magnified image. A compound microscope with an eyepiece containing a single set of lenses is monocular. One with an eyepiece containing a double set of lenses is binocular. Just above the stage is the nosepiece. The nosepiece contains the objective lenses, which are the microscope's main lenses. A compound microscope usually has 3 to 5 objective lenses, each with a different magnification power. Each objective lens snaps into place when the nosepiece is rotated to the proper position. The shortest objective lens is the scanning objective lens, which has a magnification power of 4x. A 4x magnification means that the lens is capable of magnifying an image 4 times as large as the actual object. The low-power objective lens usually has a magnification power of 10x. This microscope has a medium-power objective with a magnification power of 20x. The high-power objective lens has a magnification power of 40x. A lab-grade compound microscope will also have a 100x or 200x objective. This microscope has a 100x objective lens. To calculate the total magnification of a microscope, multiply the power of the objective lens by the power of the eyepiece. The eyepiece usually has a power of 10x, so if we are using the 40x objective lens, the total magnification of the image will be 400x. The stage of the microscope is the flat surface that holds the specimen to be viewed. The stage has a hole in the center called an aperture. The aperture allows light to reach the objective lens. Our microscope has a mechanical stage that allows the viewer to move the specimen forward, backward, and from side to side with 2 stage adjustment knobs. Just behind the stage are the focus adjustment knobs. These knobs move the stage up or down to bring the image into focus. In some models of microscopes, the adjustment knobs raise or lower the nosepiece and head instead of the stage. The larger knob is the coarse adjustment knob, used for approximate focusing. The smaller knob is the fine adjustment knob, used for sharp focusing. The base of the microscope contains the light source. Some compound microscopes use a mirror to focus light from an outside source up through the aperture in the stage. However, most modern compound microscopes have powered light sources. Specimens to be viewed with a compound microscope are placed on a rectangular piece of glass or plastic called a slide. A slide is always handled by its edges to avoid transferring oil or dirt onto the slide. Before placing the slide on the stage, we use the coarse adjustment knob to move the stage to its lowest position. Now we place the slide on the stage. This microscope has a mechanical stage, so the clips will automatically snap to the slide to hold it in place. If the microscope had a simple stage, we would slide the stage clips onto the slide to hold it in place. Next, we center the specimen over the aperture. Now we are ready to switch on the light source. If we had a simple microscope, we would adjust the mirror so the light is focused through the aperture. With the scanning objective lens in line with the aperture, we use the coarse adjustment knob to move the stage up as high as it will go. Most modern microscopes have a stage lock that prevents the stage from being raised too high, but some older models do not have this feature. We must never allow the stage to be raised so high that the slide touches an objective lens. This could damage the slide or the lens. With the light on, the specimen centered over the aperture, and the stage raised, it is time to focus the image. While looking into the eyepiece, we turn the coarse adjustment knob to lower the stage until the image comes into focus. After focusing the image as much as possible with the coarse adjustment knob, we use the fine adjustment knob to make the image sharper. With a good quality microscope, we should be able to switch to a higher magnification without readjusting the focus. To do this, we turn the nosepiece until the low-power objective lens snaps into place. If any focusing is necessary, we turn only the fine adjustment knob until the image comes into focus. To view the specimen with a higher power objective lens, we follow the same steps. However, when switching to the higher power objective lens, we must make certain the lens does not touch the slide. When we finish viewing a specimen with the microscope, we lower the stage to its lowest position and remove the slide. Before putting the microscope away, turn off the light and unplug the electric cord. If you need to transport a microscope, lift it by the arm and place a hand under the base to support the weight of the instrument. A microscope is a piece of precision equipment, and if used properly, it can greatly enhance our study of biology. In our next lab, we will continue to work with the microscope as we begin to study microbiology. At this time, proceed with the corresponding activities.